Today I'm excited to talk about the science behind 4Life Gold Factor. As you learned from the general session 1, Gold Factor serves as an activator for cells, providing a burst of power when and where it's needed. It is a delicate suspension of pure, ultrafine, crystalline gold particles uniquely developed and exclusively licensed to 4Life. To begin, let's appreciate that the human body contains more than 37 trillion cells. To put that in perspective, that number is at least 800 billion times more than the number of identified galaxies within our universe. Human cells come in all shapes and sizes, working together much like a symphony to carry out your body's everyday functions and needs. Each one of these trillions of cells requires energy to perform those functions. Each cell can be viewed as a tiny biochemical factory performing many millions of reactions every second. One of the most important of these cellular reactions is the activation of NAD+. NAD+, also called nicotinamide adenine dinucleotide, is a coenzyme or molecule found in all living cells and is activated through the conversion of NADH. It plays a vital role in energy metabolism and helps maintain proper cell function. NAD+ is central to many aspects of health. This is where the science of 4Life Gold Factor begins. There are approximately 792 billion gold crystals suspended in every milliliter of Gold Factor. Each gold crystal in Gold Factor can take on 4,600 electrons per second. This means each crystal can activate several millions of reactions on its own, which translates into trillions and trillions of cellular reactions per ounce. And because Gold Factor serves as an activator for these cellular reactions, it can remain available for many cycles. Gold Factor is uniquely developed and exclusively licensed to 4Life, and while it is highly complex and cutting edge, it is an elegantly simple process that creates precious, pure, and powerful gold crystals. Ultra-pure water is placed in tanks called troughs. This water is completely free of any contaminants and is the same type of water that is utilized in the semiconductor industry to make the computer processors used in your cell phones. Highly pure gold electrodes are submerged in this ultra-pure water. The electrodes are 99.99% pure, which is 10 times purer than 24-karat gold. The electrodes are paired with specialized equipment that controls in a very precise manner the electrical current that is applied to the electrodes. Once the controllers are turned on, tiny gold crystals are formed in the trough. Over a specified amount of time, these crystals become more concentrated. And at the end of this process, Gold Factor is created and collected into separate containers that are later packaged into our distinctive Gold Factor bottles. The tiny gold crystals found in Gold Factor are very small and come in many different sizes and shapes. We can measure the size of the crystals using a technique called transmission electron microscopy, or TEM. This state-of-the-art technology allows us to see the varying sizes and how evenly they are distributed in the ultra-pure water. On average, the size of the crystals is between 10 to 30 nanometers in diameter. So how big is Gold Factor crystal? What is a nanometer? Well, as you can see from the size comparison model, Gold Factor crystals are indeed very, very small. In fact, they're generally between the size of an antibody that might be used in a vaccine and the size of a virus, which that vaccine might be used against. And to put that into perspective, it is about 10 million times smaller than a baseball and 1 million times smaller than a period at the end of a sentence. Now let's move to a different area of health, but one that also benefits from the ability of Gold Factor to increase available energy in our body cells. To begin, we enlisted the expertise of Neurosys, a leading independent laboratory specializing in preclinical neuropharmacology modeling located in the south of France. Our collaboration encompassed direct and thoughtful interactions with Dr. Noel Calazat, the co-founder and chief scientific officer of Neurosys. In these studies, we wanted to answer the fundamental question, how can Gold Factor support neuronal maintenance, or in simplest terms, brain health? Maintaining your brain cells is one of the most energy-consuming processes your body carries out. So it makes sense that Gold Factor, which serves as an activator of cellular processes, might have a positive effect on neuronal maintenance or brain health. Why is neuronal maintenance so important to the brain? Neurons involved in many complex and essential brain functions. They are the only nucleated cell that doesn't multiply over our lifetime. Neurons are also extremely fragile. They are sensitive to many different biological stresses, and they can die. One of the greatest, most recent neurological discoveries is that of neuroplasticity, which is the ability of neuronal networks in the brain to change and improve through growth and reorganization. The significance of this discovery is that neuroplasticity is not static and can evolve from the beginning until the end of human life. As a result, improvements in mental acuity and memory, how we think, how we learn, perceive, and remember are all possible through brain exercise, or perhaps even through supplementation. In these experiments, brain cells were grown for 3 days and then divided into different groups. Each group was treated separately with and without gold factor for 7 or 10 days. For 7-day experiments, brain cells were grown under normal conditions. For 10-day experiments, brain cells were deprived of growth factors, which help imitate an accelerated aging process. After the treatment period, the brain cells were collected and stained with fluorescent dyes to aid in the detection of neuronal networks. High-resolution photos of fluorescing cells were captured with next-generation, high-content imaging system that incorporates machine learning as part of its processing. These processing techniques were used to analyze neuron survival, neurite network, and the number of synapses in the brain cells. As you can see from the chart, under the conditions of normal brain cell growth, there was a significant improvement across all measures of neuronal maintenance. That is demonstrated by an increase in the gold factor treatment compared to the no treatment group. Under the conditions of deprived brain cell growth, there was a significant improvement across 2 of the 3 indicators of neuronal maintenance. In the context of this experiment, when compared to no treatment, gold factor treatment reduced the negative effects of brain cells that were deprived of growth factors. In summary, gold factor increases the number of neurons, grows the neuronal network, and increases the number of synapses under normal conditions. Gold factor was also able to protect the neuronal network under accelerated aging conditions. Together, these results confirm that gold factor holds the strong potential to improve neuronal maintenance, which helps protect and boost mental acuity and memory. Finally, let's turn our attention to the potential benefits of gold factor as it relates to longevity or anti-aging. One of the most extraordinary investigations of gold factor was an independent study from a lab called Rosecamp Institute located in Sarasota, Florida. They are a highly specialized research lab that investigates the fundamental and complex issues that arise during the aging process. Dr. Ganya Ait Ghazala was the principal investigator for this study. She has a PhD in molecular and human genetics from the University of Paris and has studied and published research on nutritional supplements just like gold factor. One of the most cutting-edge areas of aging research is that of telomeres. And before we show the incredible gold factor research on telomeres, let's first talk about their fascinating history. In the early 1970s, Russian theorist Alexei Olovnikov first recognized that chromosomes could not completely replicate their ends. Olovnikov suggested that DNA sequences are lost every time a cell replicates until the loss reaches a critical level called the Hayflick phenomena, at which point cell division ends. Later, Elizabeth Blackburn discovered how chromosomes are protected by telomeres and the enzyme telomerase. This protection can allow cell division to continue when it might otherwise have ended. She was awarded the Nobel Prize for this most important finding. Now you may wonder why telomeres and telomerase are so important for your cells and for your body, so let's examine this a little closer. The average cell will divide between 50 to 70 times before it dies. Each time a cell divides, the ends of DNA, called telomeres, become shorter. You can think of telomeres like the end cap of a shoelace. Over time, telomeres become too short to protect the chromosomal DNA. When that happens, a cell can no longer divide and it becomes senescent, which is defined as the state of being old. Telomeres essentially act as a timekeeper of the cells, and as such, telomeres can be thought of as a biomarker of aging. Telomerase is an enzyme that protects telomeres. It is a structure that has both protein, which is colored in gray, and RNA, which is colored in yellow. During cell replication and division process, telomerase binds to the overhang of a DNA strand, which is colored in green. Then it uses its own RNA as a template to extend the DNA sequences. By extending the DNA sequence, telomerase extends the telomere of chromosomes. In simplest terms, telomerase can be thought of as a biofactory that repairs the ends of cellular DNA. In this study, telomerase activity was monitored when cells were treated with gold factor and when they were not. This was done at each passage or cell division. The method can be described as such. After the first passage, cells were divided into two separate groups. For each passage, each group was treated separately without and with gold factor for around 72 hours. Cells were collected after specific passages and frozen and stored for analysis later. Measuring telomerase activity is a complex multi-step procedure. The first steps involve magnifying the portions of telomeric DNA so that they can be detected. The final steps utilize a process called ELISA, which stands for Enzyme-Linked Immunosorbent Assay, and is a method that allows for detection of very small amounts of peptides and proteins like telomerase. In this process, a detection molecule is added to the telomeric DNA, which makes it turn blue. The blue-colored DNA portions are then detected based on their intensity, which is proportional to the amount of telomerase in the sample. The first set of experiments was done as a proof of concept. It was based on the premise that gold factor may activate telomerase to help slow down telomere degradation. In the next few slides, you will see that the proposed mechanism for this hypothesis. Let's first examine this chart in detail. On the left half are the cells with for-life gold factor treatment, and on the right half are cells with no treatment. Different colors of the bar indicate different passages. For example, gray is for passage 2 and purple is for passage 15. The heights of the bar indicate the activity of telomerase. All the bars have been normalized by comparison to that of passage 2. For the cells with no treatment, telomerase activity gradually decreased as the cells divided. Passage 2 has the highest telomerase activity, passage 8 is lower, passage 12 is even lower, and passage 15 has the lowest telomerase activity for cells with no treatment. And as you already know, this is part of the normal aging process as cells divide. For cells with for-life gold factor, in contrast, telomerase activity showed a trend in the opposite direction of the no treatment group. That is, a gradual increase of telomerase was observed as the cells divided. So passage 8 is higher than passage 2, passage 12 is even higher, and passage 15 has the highest telomerase activity with gold factor treatment. In a follow-on study, we examined gold factor at several concentrations and compared it to other well-known anti-aging and telomere-supporting supplements that are currently for sale in the marketplace. The charts are represented in a similar manner as before, looking at telomerase activity of each passage with each different treatment. And if you focus on the right of the chart, you will see that gold factor again shows an increase in telomerase with each passage. Most interesting is the significant increase in gold factor's telomerase concentration compared to the other leading anti-aging supplements. It shows the greatest increase in telomerase concentration. So how exactly does gold factor activate telomerase? It's still too early to know for sure, but we have an idea that can begin to help us understand the impact of gold factor as an activator for cells, providing that energy when and where it's needed. And it starts with the NAD cellular reaction that we talked about earlier, to which gold factor serves as an activator. In our cells, there's a protein enzyme called tan-chyrase, which requires NAD for activation. Tan-chyrase works alongside telomerase to help protect and lengthen telomeres. It does so by blocking a protein called telomeric repeat factor 1, or TRF1, which binds to the telomeres and prevents them from lengthening. So gold factor activates tan-chyrase and telomerase through the NAD reaction, which blocks TRF1 and as a result helps protect our telomeres. Of course, as you can imagine, more work needs to be done to strengthen this hypothesis. These results from an independent state-of-the-art lab confirm that gold factor holds the strong potential to activate telomerase. In these studies, gold factor was able to increase telomerase activity significantly compared to no treatment, and when compared to other leading brand anti-aging supplements, gold factor performed as well or better. As you've just seen, the innovative research behind for-life gold factor is another amazing example of for-life's commitment to science. The sheer depth and number of research studies on gold factor demonstrates why science is the first of 4Life's 4 values and why research is in our name. 4Life Gold Factor indeed is a cellular activator solution, providing a burst of power to our cells when and where it's needed. It is a first-in-class product in a long line of groundbreaking, exclusive 4Life products. Thank you.